I'm going to jump up here and talk to you all just a little bit about taxonomic diversity in ash. But this is technically a co-presentation with myself, Julian, and Alan in a discussion format. So do expect me to be bouncing back and forth between all of us to answer specific questions. I am coming from this more from the genomics and bioinformatics perspective, looking at basic biodiversity to understand what kind of ash we have on the landscape. And so, yeah, again, I'm Zane, if you didn't meet me yesterday. What? Oh, sorry. I'm Zane Smith from the University of Tennessee, and I work in Meg Staton's lab. Okay, so the first thing that I think is really important to just sort of set the stage here is that Fraxinus is a reasonably large, somewhere in the ballpark of 60-ish, 70-ish species. The actual species boundaries across the world are relatively poorly defined, and we're going to talk about that pretty extensively. Why? But I think the first important thing to know is that there are 6 major sections of ash. There's the New World dipetalae, which includes species like blue ash, Passiflora, which is mostly restricted to the southwestern US and Mexico, section Melioides, which we're going to talk about mostly today because that's sort of where Julian, Meg, Alan, and I mostly live at the moment. It's a very special section, and we'll talk about that extensively. And then we have the primarily Eurasian sections, which are section Ornis, Sciadanthus, and section Fraxinus. Notably, black ash here from the United States is also in section Fraxinus. So we have this beautiful rainbow of species. But what's really important evolutionarily, I think, to consider in thinking about how we seek to frame and conserve these species are that not only do we have broad species diversity, but we also have broad evolutionary mechanisms within the genus. So we have rampant self-incompatibility, and we also have a lot of sexual dimorphism that varies throughout the taxonomic sections. We have everything from self-incompatible bisexual flowers to completely dioecious male and female species. And so again, the section that we're mostly working in is section Melioides, and they are all dioecious. Also, super fun, we have a ton of polyploidy in this section in particular, but very little throughout the rest of the genus. But Fraxinus also sees quite a lot of cryptic hybridization on the landscape, which is going to be really important thinking about moving forward and how we think about conserving and partitioning species diversity versus hybrid and genetic diversity on the landscape. So very briefly, I took this slide from some of Alan's previous work. This is just a brief overview of some of these species within the North American sections. And I just wanted to point out here in section Melioides that we have several species that are either have been proposed in the literature but are not well accepted or not well understood, and several that are also new to science. And so these vary from diploids to tetraploids and all kinds of fun stuff. So I just wanted to throw this up here just to show that we currently have somewhere in the ballpark of probably 20% of section Melioides that is not particularly well understood or described. So why are ash species hard to identify? If I took these species names off of here, I would imagine that you would all believe me if I told you they were the same species, and you would probably believe me if I told you we were all different. So you can see we've got 2 of the most common eastern species here, which are green and white ash on the far left and the far right, as well as our wonderful cryptic hexaploid, which is native to this region, Whittemore ash. Again, not particularly different looking, reasonably easy to get confused in the field. A lot of the macroscopic characters are not particularly informative of their underlying taxonomy and their underlying ploidy problems. So why does this actually matter? Well, if we think about we think about restoration breeding units, for example. It's really important for conservation to understand what we have on the landscape if we're looking to conserve it. I think we're going to circle back to that later and talk a little bit about the red listing. So I'm super excited that Kate Good is here. I think we might become really good friends. Once again, this is where we are with EAB. So largely the Southwest is currently untouched. Everywhere else is pretty much touched by EAB. The main species that are seeing a lot of ongoing conservation action at the moment are green, white, and black ash, and as well as Oregon ash now, thanks to Richard Sneezco, the folks in Oregon. But largely these other species have been sort of swept off to the side as we'll do the best we can, we'll see what we can do, but I think that we can do better. So I've mentioned polyploidy, and I just wanted to sort of recap what that is, what that means, and why that's important. So there's a couple of kinds of polyploidy, but so to start off with a human example, we are all diploids. So we each have a copy of our genome from mom, a copy of our genome from dad, and many other species are the same way. However, plants are reasonably tolerant to having more than 2 copies of their genomes. And so you can see here in the bottom left, this is a triploid. It's a plant that has 3 copies of its genome. Tetraploid has 4, and in ash we go all the way up to 8. So it gets pretty messy pretty quickly. But this is just assuming that your genome is doubling and doubling and doubling within a single species. So that's one mechanism. A more common mechanism is you have hybridization. Again, we see this in ash frequently. So you have one species and another species, they hybridize, you end up with 1 copy of the genome from one parent, 1 copy of the genome from the other species parent, and then you have a duplication event, and then you have this awesome mosaic of diversity from multiple species. So you might be wondering, well, which one of these do we have in ash? Why do we really care about this? Why does this matter? This is really important for thinking about, number one, genetic novelty. So this is a really valuable way for genes to take on new functions and do new things. So when we're thinking about resistance to emerald ash borer, for example, thinking about how defense genes evolve and expand or potentially contract in some cases is important to understanding the evolutionary history and also the evolutionary future of Fraxinus, really across the world. So which one of these do we see in ash? Well, we don't know, but the answer is probably both and probably sometimes a mixture. There will be more to come on that in the near future, but I'm happy to talk about it in the discussion. And I know Liz asked us to do this up front, totally forgot. If you have questions, please, by all means, feel free to raise your hand, or we can wait till the end, totally up to you all. I don't mind a bit. I wanted to throw this wonderful sort of tweaked figure from the paper so you all can read it. This is from Alan Whittemore and Julian Campbell's 2018 paper, Ploidy Variation in Fraxinus, looking at the different ploidies throughout North America in this suddenly endangered genus, which is my favorite way to refer to Fraxinus. So what you're essentially seeing here is that under the 2, a lot of these individuals that were sampled are diploids. They have 2 copies of the genome. This is just based on flow cytometry, which is a nice molecular way to validate how big your genome size is, and then essentially extrapolate that to how many copies of the genome that you have. The summary, I guess the major summary of this figure, if you start looking at this, I don't know if I have a pointer here. I do not. I do. Right here, we start seeing our tetraploid, 1 pentaploid, hexaploid, and also some octaploid individuals down here. But you see that across these different botanically assigned species IDs, this wasn't always holding up. There was a decent bit of confusion. And so this really, this spurred a lot of efforts between our lab, Julian, Morton Arboretum, to do some additional sequencing to try and understand what's actually happening on the landscape and where we actually start to delimit these species. So I'm going to talk to you very, very briefly about some partitions of the North American Melioides ashes that I find helpful to sort of frame the diversity in my mind. These are not all 100% phylogenetically accurate, but I think that you might find it useful. So I want to start and just talk very briefly about the green ashes, which we see here largely in the eastern United States. One of the best characters that we have found for identifying members in this group. So this is green ash, Fraxinus pennsylvanica, Rio Grande ash down from the Texas-Mexico border, as well as a potentially new species that appears to hybridize quite rampantly with green ash. We've been calling it Fraxinus campestris, but that is a story for another day. I think what's important to know is that one of the most popular cultivars of ash that have been planted throughout cities across the US is Summit. So Fraxinus pennsylvanica Summit, and it actually falls into this Fraxinus campestris group. So we're moving around cryptic diversity in our urban landscapes as well as in the wild. So in terms of identifying, is something a green ash in the broadest kind of sense? This is a terrible photograph that I took through my phone with a little cheap $10 hand microscope of the bottom surface of the leaf. And you'll see that this is just relatively smooth and shiny looking. What you're seeing are just different cells here and just relatively smooth surfaces. This is important when we start thinking about the white ashes, where you can see they have these nice little papillose cotton ball-like structures that reticulate on the bottom. And this is one of the best ways to just immediately tell, am I dealing with green ash or white ash on the landscape, or something more likely within one of those two groups? So on the left here, again, this is a photo of my cheap microscope. I use this really often in the field for surveying, just to get a better idea of, am I looking at green ash? Am I maybe looking at something a little bit more cryptic in the white ash complex? Because we have these really broad phenotypic gradients that all sort of mix and overlap. So there's really not a one good character for splitting out these species. You really have to look at 2, 3, 4 to get a consensus. So in the white ashes, we have several species. We have Fraxinus americana, white ash, Fraxinus albicans, which is endemic to Texas. We have a new species that we're happy to regale you about a little bit later, Fraxinus albopalustris, which is a tetraploid. Many of you may be familiar with with Fraxinus smallii, which was a species described by Neeson. We have essentially found, well, not we, Alan and Julian have found that this species doesn't really hold up. It's essentially the original type material are actually hexaploids, so they are Biltmore ashes, which is a good species. And so we see these mainly across the eastern United States. One species complex that is often really ignored, and it's great to be here in North Carolina because we have quite a lot of the water ashes, which technically do belong more to this larger green ash species complex that we have going on. They tend to be, an emphasis on tend, to be slightly smaller, scrubby, swampy kinds of trees, like you can see here in the top right. Samaras are a wonderful way to start getting a better idea of what these species might be. But once again, these species are dioecious. So best case scenario, you only have half of the individuals in a population that would actually have this important character. So this is where it gets really, really tricky. Although from some of our recent phylogenetics work, we've also found that pumpkin ash here, Fraxinus profunda, are wonderful octoploid, probably some sort of older hybrid allopolyploid. However, it does seem to have some affinity with this water ash complex in some way. So I've included it here. And if anyone's ever seen it, this is quite a large tree. You can see some leaves here that are essentially right about as big as one's forearm. And the Samaras tend to be 1.5 to 2 inches long, really large. Finally, just to touch on some of Alan's work really briefly here, we have the Western ashes. So this includes Oregon ash, Fraxinus latifolia, as well as velvet ash from the southwestern United States. A often confused species, Fraxinus papilosa, often conflated with Fraxinus volutina, as well as Fraxinus udii, which is really widely planted across the tropics, globally, actually. And this is our other wonderful hexaploid species that we have in the genus. Out on the landscape, though, here in the Los Angeles, California area across the Southwest, we see a lot of species that, or a lot of individuals that seem morphologically like they would belong to this Fraxinus volutina complex. However, we see diploids and tetraploids scattered across the landscape, sometimes commingling, sometimes not. And yeah, so we have more work to do in the Southwest for sure. Really quickly, just to show you all a little bit more about Fraxinus albopalustris, which there's a wonderful little site of an hour-ish or so from here. We've been talking to Adam quite a bit about this species, about trying to get some individuals planted out and growing in conservation collections, which is really exciting. But here's a little bit of the morphology, which if you're familiar with ash morphology, I'm not even bothering to show you the leaves because they're so polymorphic. They often end up being more confusing than anything else. This species has an affinity with the white ashes. We currently consider it to be part of the white ash species complex, but it's very likely allotetraploid, so of hybrid origin. I know there's been some interest from some of you about seeing just a little bit of the morphology. This is it, and I'm happy to share some of this information with you all later. This is just a general map here of our two southeastern tetraploid species produced by Julian. So in the dark black triangles, you have good, confident locations with accompanying flow cytometry data to quantify their ploidy of Fraxinus albopalustris all throughout the southeastern United States. And then you see the more traditionally recognized Fraxinus passiflora, which is, again, is a member of the water ashes that clusters here more toward the coast and into Florida largely. So we do see some good biogeographic separation in these species. Finally, I just wanted to show you one very small picture of some of the genetic work that we've been doing. Happy to talk about it later. This is just a PCA, but what I want to show you here is on the left, you can see the Fraxinus pennsylvanica species complex very broadly, which includes the water ashes. And then on the right, you can see the Fraxinus americana species complex, which includes our new species, Fraxinus palustris. You can see that they have this very steady gradient across the PCA where they do segregate genetically. We have a lot more work done on this, a lot of stuff coming out, a paper coming out very soon. Yeah. Just some other very lightning quick synergistic efforts that are going on in sort of the realm of genetics. We've also been assembling a bunch of reference genomes for all of these species, as many as we can. We currently have a male and a female green ash, Fraxinus pennsylvanica, male and a female white ash, Fraxinus americana, a female Fraxinus caroliniana. Thank you to the Sarah P. Duke Botanic Gardens for contributing that individual. We have a male and female Fraxinus manchurica. This is important for resistance breeding because this is one of the ancestral hosts of emerald ash borer. We do have a new Fraxinus nigra genome, so black ash, blue ash, Fraxinus quadrangulata. We're working on a male and a female Fraxinus biltmoriana, so we've got a couple of hexaploids coming down the pike. We're also collaborating really closely with the Wegerson Lab at the University of Connecticut to try and assemble a pumpkin ash, Fraxinus profunda. Just really quickly, this is always a question when it comes to genetics. Who cares about a reference genome? Why do you need that? Why does that matter? The way that I like to think about it is by producing a reference genome for these species, we're essentially creating a backbone, a really high quality, as complete as we can get backbone of an example genome from one of these species, so that when we go out to the wild and we think about population genetics and what does diversity look like range wide, we have something that we can take all of that additional sequencing from. That's just a little bit cheaper. We can pile it all up together and we can start to scaffold and understand how diversity is partitioned across the genome. We can also look at things like resistance genes and all of that with higher fidelity by having these reference genomes. We've also used this recently. This is work that will be published here this year in the near future to understand the genetic basis of self-incompatibility. This is really important. So we have located, much of this work was spearheaded by some folks from France, but we found the self-incompatibility locus that's ancestral to Fraxinus in our black ashes and blue ashes, as well as some of the others from Mexico as well. So that does suggest that even our bisexual species do have different compatibility groups. And we've also mapped the sex-determining region for our species in section Melioides, so Oregon ash, Carolina ash, white and green ash specifically. So this is really important for thinking about species that take 7 to 15 to 50 years sometimes to mature. We now have a way to sequence them and identify what other individuals they're likely to be compatible with when they come to maturity from the seedling stage instead of waiting until they're adults. And finally, I just wanted to throw this up here now that I've sort of shown you all of this other diversity that is poorly understood within Fraxinus, and say this is the current red list, which was an incredible and amazing effort that we've benefited greatly from. But if you just take a peek, so this is from North America specifically. We've got about 16 species here, and in North America, I believe we're up to something more akin to like 22. So we're missing a decent amount of the diversity in our gap analyses. And so I'm really, really excited to talk to all of you about how we can work together to update the red list and how we can make this maybe one of the first major products for this global conservation consortium. I think that there's a lot of utility to this. So I believe that's all that I have. And I'd really love to open it up to discussion and hear thoughts, questions, uncertainties around the diversity that we see in ash, or questions on. Go ahead. Yeah, if you have a question, please.
This is not at all my area, but thinking about where the emerald ash borer comes from, I'm sure you've thought of this. If it's Asian, are you looking at Asian ashes?
Yeah, so this was a big reason that we sequenced the genomes of Manchurian ash. So the Northern Research Station has been doing some hybridization between black ash and Manchurian ash. It's super complicated because they have different sex systems and all kinds of fun stuff happening there. But there is some ongoing interest in thinking about, can we try and do like what American chestnut has attempted to do and get some of this resistance from the Asian sources and introduce it to.
So I was just checking with our Asiatic curator to see how many ashes he's got. He's got 4 different species in 2 gardens.
Amazing. Okay.
So if you're interested, I'm sure he would be able to help us to you.
Yeah, I would love. I'll follow up after. I'd love to talk more about that.
Most of the varieties do not have an Asian.
Yeah.
And most of our North American ashes, that's not.
The species that's come from, well, called Staton, there's some. The Asian ashes are not a clean taxonomy. Just leave it at that.
Nothing is.
But yeah, so not across the Pennsylvania, we have 1 male Staton. We are able to use that pollen on, grow trees, we run it through the bioassay, we're cutting up them. So it's cross compatible. Everything else we tried with pollen outside of Manchurian, all of the orange group doesn't seem to be compatible. So, and that's not exhaustive. We puttered around with it. Before lingering ash was identified, we thought that a hybrid approach was going to be the way. That was the initial sort of plan. And then once we realized we worked within the best, shifted our effort. So, but it doesn't look like there's good partners there. Of course, we never got into China. So.
Great question. Chisenensis from
That
Korea.
That might also be.
super quickly, Mary, so the folks online can't hear super well. I apologize.
Yeah, so we have these little microphones, and my understanding had been that they would sort of capture the room, but I guess we do need to be better about just making sure they're near the people who are talking, so I can shuttle this around as people from the audience talk.
Great. I'm going to summarize really quickly and just say that we had a question about, are we looking at the Asian ashes, so the ancestral hosts of EAB, and thinking about potentially being able to move resistance? Are we looking at any of that diversity there? The answer is yes. And Mary was explaining that in a couple of instances, the Northern Research Station Ash Restoration Breeding Program has done some crosses both with among black ash and Manchurian ash, as well as green ash and Fraxinus platypoda spathiana. It's one of those poorly kind of delimited species. And so that was sort of just the ongoing discussion there. What other questions do we have? Kathleen, please.
What do we know about the native rain?
Very good.
What do we know about the native range of Fraxinus profunda? I think it's one that's often confused with green ash and is a little bit cryptic in the landscape. So that seems like a challenge.
Yeah, so what do we know about the range of Fraxinus profunda? This is a great question, and I'm going to give you an even broader answer of that. What do we know about the ranges of any of these cryptic and confusing species? And that is not very much. Fraxinus profunda specifically has this odd relict sort of ring range from Canada all the way down to Louisiana, I believe. But again, we have also just all of these other species, these cryptic polyploids like Fraxinus biltmoriana, Fraxinus albopalustris now, our tetraploid volutinus from the Southwest, that we just don't have an amazing grasp of what the exhaustive ranges are of those. And so this is, again, something that I'm really glad you brought up, because would love to talk to more folks in this network about trying to do a better job of getting out in the landscape, figuring out which features are important to delimit these so that we can establish better range maps and create a better scaffold for surveying genetic diversity, thinking about novel sources of resistance potentially as well. Please.
We have an online question. It's in the chat, so I can read it.
Okay.
Isabel and Natalie say, thank you for the excellent presentation. You put a lot of effort into distinguishing species for conservation purposes, but they would like to hear your thoughts on the evolutionary potential of the genus, especially with mechanisms such as polyploidy and hybridization.
Yeah, this is great. And I don't want to steal Mary's thunder here later on. I think that she'll have some much more interesting thoughts on this than I will. But I think it's important to consider what ideal versus realistic is when it comes to conservation and breeding. I think we're still at a stage where all hope is not lost for ash. Many of these species are still almost untouched. They're just not by emerald ash borer or barely touched. They're just not well understood. So we have a lot of opportunities now to get out in the landscape and try and understand them better before they become severely impacted, like some of our more northeastern species. But I think it's important in some cases to think a little bit more broadly about what we consider as conservation units. So, for example, in areas of the landscape where we do see hybridization between some of these species, I think there's value to that. That's a natural, ongoing ecological and evolutionary process. I think that there's some ways that we can try and strategically, hopefully in the near future, move resistance around between some of these species and try and do some back crossing in order to try and maintain some of the historical ancestral traits and ecological affinities of these species, while also not fixating on purity, because species are just a useful convention anyway. Biology doesn't really care so much about our boxes. What other questions do we have, or how are we on?
We are ready for the next session. Actually, wait. Nope, I'm wrong. We have 15 more minutes.
Okay.
Sorry, I had it totally wrong.
Really quickly, Alan or Julian, I've taken up most of this time sort of blathering on. Is there anything that you all would like to add?
I could pull some of the type material onto the table here to show people some real specimens in a bit. I'll do that in a while.
That would be incredible. So Julian is going to actually, he's brought some Julian has brought some physical specimens of this new species that we're working on describing here for folks in person to see. And I'm sure we could circulate some photos or add them to the Padlet later so that people can get an idea of what we're talking about morphologically with this species.
Yeah, maybe during the break, you can lay those out, Julian.
So it seems that there's not a 4N species in the pen group, in the green ash group.
Yeah, this
So that's as much as we've got out there, and is probably you might imagine that to be the easier polyploid to make. So that's, what do you think? Are we missing a species there still?
That's a great question. I think that there's a reasonable chance okay, let me back up. I know that we don't have an exhaustive understanding of diversity on the landscape. I think anyone who's spent any amount of time in the field just looking at ashes would probably agree with that statement. There's a lot of maybe this is different, maybe this is similar. But using some of the genetic techniques, I think that we can get at that a little bit better. But in terms of specifically a tetraploid green ash type species, I think that we potentially see that in Fraxinus albopalustris. It does morphologically cluster with the white ashes, but in our upcoming manuscript, we'll describe a little bit more about how it appears that it may be a hybrid between a member of the broader Fraxinus pennsylvanica green ash species complex and a member of the white ashes. There's still significant ambiguity there. It's a question that we're actively chasing. But yeah, I think it's possible, but we don't know. Kathleen?
So just following up on the pumpkin ash, if anyone is working on kind of a range map or anything with it, I had assembled some different published and gray literature sources on range maps in my region, and also added a bunch of counties that I found pumpkin ash and have herbarium specimens and seeds from those at our lab in Delaware. So I'm happy to work with anyone who's interested in that information.
That's absolutely amazing. I'm so glad to hear that. I definitely think that species needs a little bit more attention. I haven't actually mentioned this yet, but we do have some early evidence based upon what's called RADseq, which is just like sequencing a small portion of the genome, more or less. That pumpkin ash also may not just be one taxonomic unit. There might be a couple of species mixed in that pumpkin ash group. More to come on that in the next couple of years, probably, but we'll have some more sequencing information on that here probably in the next few months. And we'll definitely keep the group updated on what we find. I think it's really important to try and communicate these early findings on potential updates to the taxonomy to stakeholders, boots on the ground, as soon as possible. The folks can just keep an eye out and sort of reframe maybe the way that they're looking at some of these species. Other questions, please.
My question is kind of centered around the cost of sequencing. In mushrooms, there's DNA barcoding. Are we at that stage in ash where there's a very cheap sequence we can run on a specific portion of the genome that's very affordable? So say people who are collecting can afford to just send in samples and pay to get them tested to verify what they're finding?
This is a wonderful question, and I'm really glad that you asked it. So there's several layered answers to this. The first one is that Richard Buggs's group from Kew recently published a paper on potential genetic barcodes to try and delimit some of these species in a more cost-effective way. The plan is currently to take a lot of the whole genome sequencing data that we're generating and evaluate how well these markers perform in individuals that we have ploidy information for, whole genome sequencing, botanical information, or so morphology from Julian and Alan, to just see how well does this hold up in general, how well does it hold up in the North American species in particular. And so the short answer is we're not there yet. We don't have an amazing cost-effective way to sequence and identify them. I think the base cost is still somewhere in the ballpark of $35 to $40 at scale to try and identify some of these things. So it's not very easy to do a one-off, but maybe that's something that the group could consider discussing in the future. If people have a lot of ash diversity on the landscape, maybe there's a way we can coordinate, potentially pool funds across groups and try and, get a sequencing plate into a company and then try and identify some of what's going on in different regions and individuals of interest. So it is an area of very active interest. We're just not quite there yet. Go ahead.
The mechanism of resistance. I mean, is there any resistance mechanism? Is it on the sort of the laying of the eggs? Is it the feeding? Is it the taste? Is it are people looking at all of that?
This is a great question. Yes, people are looking at all of this. So our lab is working very closely with the restoration breeding program. That is Meg and I. Try and understand more about emerald ash borer resistance. So my understanding, Mary, you can jump in and correct me if I'm wrong at any point, is that there are two major-ish kinds of resistance, it seems. One that is more focused on, actually, maybe Mary, you can speak more intelligently about this. Just
Well, of course, there's a lot of unknowns. EAB is intimately associated with ash throughout its entire life cycle. The places we've been able to look at and kind of quantify or measure, do an experiment on that interaction is adult. We did some adult landing and feeding studies, or I say we, Therese Poland's group did some landing and feeding studies early on, and with I think 4 or 5 lingering ash are the earliest selections, and there are differences. Likely based on differences in volatiles, and that may be what's going on with blue ash. It's got very different volatiles. It may be harder for EAB to find. So there's definitely some things there. Therese tried some egg laying preference studies, and the EAB are like incredibly uncooperative. They would rather like die than lay an egg on a tree that you gave them. So, what has worked really well is transferring eggs to trees and seeing what happens to those eggs. And I've got a little bit of that coming up on my talk later. But it's definitely true that many lingering ash kill more EAB larvae than unselected trees. And the earlier you can, knowing how the damage happens, obviously the earlier you kill a larvae, the better probably your health is if you're an ash tree. So there may well be other, so we don't have enough data in from the field yet so not all the lingering ash do well in the egg transfer bioassay. So I suspect that there are some other places and that resistance and some other mechanisms, and that as we're able to get our field data in, that we may see have a better idea what some of that is. But as the geneticist on the breeding program, I'm always reminding folks, I don't need to know why, I just need to know who's a good tree. For the breeding. Obviously, as a scientist, I'd love to know every single gene involved and exactly how it's working, but we have, yeah.
You want to be able to select when you don't have a forest of genotype trees already. It really helps if you can say, okay, this is a critical gene, and pick out the seedlings that have favorable resistance genes. So it can be very helpful. Otherwise you wind up like, the attempts to resistance from Asian chestnut into Castagna dentata, which worked, but you know, they started them around 1920 and they were getting trees in the ground in the late 80s. So you prefer not to take that long.
There's, I mean, obviously a really good marker. There are huge opportunities and there's numerous examples from crop systems where good markers have accelerated breeding. So I'm not, I'm not against that, obviously. But, we bred almost all the food we eat was bred for thousands of years before we knew what DNA was. So, I mean, do you have to have it if there's only so much money and you have to choose? We can't do everything. How do you choose what to do? That's not a question I'm going to answer. That question for the community to answer. But yeah, I think it's.
And one other thing I want to mention here too is I've largely framed this as some of the work that myself, Julian, Alan, and Meg have been spearheading. But I do also want to make it super abundantly clear that we have something in the ballpark of 20 to 30 partner institutions working on all of this with us. That includes Adam here at Bartlett, the university, the National University in Mexico, Natalie Isabel's group at the Canadian Forest Service, many folks from out west, botanical gardens, including Sarah P. Duke, Naples, Lady Bird Johnson Wildflower Center, and many, many, many more. We've been casting a really broad net from institutions to personal individuals to try and at least establish a reasonable baseline for what we see on the landscape with just a humongous network of people. So I also don't want to, just want to make that clear that this has already been a really large community effort from all sorts of individuals and institutions, and we're really excited to try and hopefully work with the rest of you as well. So. Good.
Got 3 minutes, so good transfer time.
Great.
Thank you, Zane. See if there. Okay, so next up we've got Meg and your lingering ash.